Good afternoon, everybody. We took a few minutes more to let everybody join. So now we are going to start with the third session. It will be about EPTRI, networking, outreach, and development. So it is a pleasure for me to introduce the first speaker, Mariangela Lupo from the TEDI Network. She will have a presentation on EPTRI outreach and networking plan. Please, Mariangela.
So thank you, Donato, for welcome me. Okay, so I would like now to present which is the global outreach and networking strategies that we have defined in order to help EPTRI to liaise with all the many international pediatric initiatives that, as has been already underlined, but all the other colleagues from Mark, from Donato also, and from Pirko as well, it is really important to be included in the general framework and to try to build links and relationship with all the initiatives in the field that we have to link with. Therefore, we have defined an outreach and networking strategies, which has the main goal to first increase the impact of EPTRI services and initiatives, but also its global partnership in order to also reduce the barriers between the basic science and citizens' well-being, providing also evidence of the direct benefits that EPTRI can have onto the society, but also to link and to liaise with the many pediatric initiatives which are conducted both at national and international level, but also with the international research programs and projects. Key to the success of these activities will be the critical involvement of the patient community and the building of liaisons with all the relevant stakeholders that include the scientific community, the policy and regulatory decision makers, but also the pharma and biotech industry and the SMEs. In particular, we would like to set up an external stakeholder forum, which would have the aim to promote the much needed research in the pediatric population with special attention to the unmet need areas like the rare diseases, neonates, and rare cancer, but also to leverage the existing initiative, project, and knowledge in order to keep the pediatric medicines development high in the European agenda. Next slide, please. In order to set and to properly identify the activities that have to be tailored to the different target audience, we have identified which are the addressees of our activities. So first of all, the principal target that our activities will be addressed are for sure the potential infrastructure users that can come from both public and the private sector, the biomedical pediatric research and development community, the relevant ESFRI and other world-class research infrastructure and networks, including also the European reference networks, the pediatric biopharmaceutical and medical device industries, SMEs, CROs, and startups, but also the patient organization, young person advisory group representatives, citizens, and the governments, the European body, the regulator, policymakers, and funders. In order to maximize the outreach and networking strategies, we will set up different and coordinated communication activities, but also we will develop a comprehensive set of tools for the specific target audience according to the different channels, and implementing also as well informative materials to promote the dissemination activities. So next slide, please. Okay, so the first set of activities that we will perform will be addressed to the governments As you know, during the design phase, EPTRI has already received several support letters from the government, which have recognized the value of EPTRI in enhancing the pediatric research. As EPTRI main goal is to become a European research infrastructure consortium upon reaching the fully operational stage, it needs for sure to receive support from the governments of that participating country. To the same, a coherent and strategy-led approach to policy makers, both in European and non-European countries, will be developed, not only to receive expression of political support from those governments, but also to let EPTRI be included in the national funding and in national roadmaps. To the same, so several meetings and tailored activities will be organized at the governmental level to negotiate the support to the EPTRI to the future EPTRI research infrastructure. So in order to making the funding bodies aware of the strategic funding needs of the pediatric scientific community, and also to convince them to establish long-range plans and roadmap, we have planned and we have already performing this kind of activity. So some measures have been defined. So first of all, we have focused on the identification of which are the relevant stakeholders to be contacted in each country, in order so to spread the word about EPTRI, its mission, our pathway, and to start a collaborative work. Moreover, we have involved the regional and the local authorities in order to enhance the possibility of benefit of the European Structural and Investment Fund of other financial support facilities, both at national, local, and European level. And the previous presentation at the end of last session was really a very good example on how activities at local, regional, national level have been performed, and we are trying to strengthening these activities. Then we have also, and we will continue testing the interests of the governments on setting up a research infrastructure dedicated to pediatric research through interviews, focus group, and short questionnaire, but also linked to the organization of face-to-face meetings on teleconferences with the main identified stakeholders. So both from the coordination team, the relevant national authority representatives, the coordinator of the thematic research platform, but also involving the representative and the coordinators of the national nodes in order to identify and discuss with them which is the possible type of involvement. And finally, it should be, and in some case we succeed in this, in the formalization of the involvement in EPTRI, and according to this, we have received several governments which have also signed so far the memorandum of understanding, and we expect that other countries, other governments would like to sign also the memorandums as well. We have received a lot of endorsement letter that would be very crucial to be submitted in the next application that is expected to be finalized on the 9th of September for the inclusion of EPTRI, hopefully the inclusion of EPTRI in the ESFRI Roadmap 2021. Next slide, please. Okay, the second target audience that we would like to, we will reach in the EPTRI phases would be for sure the pediatric research community. To the same specific actions will be planned to liaise with the related European funded projects, society, pediatric networks, ERN, and professional associations, mainly to engage with relevant communities and to strengthen cooperation with European joint programming and research initiative. To the same, we would really like to set up a working group in order to elaborate a sort of position paper on how to strengthen collaboration with the pediatric research initiatives. Moreover, we would like to organize several online consultations on specific topics in order to identify which are the expectations, the willingness to collaborate, and the actions that could be conducted in the collaboration with EPTRI. Several activities we want to promote the involvement of the pediatric research community will deal also with training activities, scouting of projects of common interest, and we would like also to plan ad hoc initiatives to address the questions of the pediatric medicines for rare diseases. And to the same, all the services available at EPTRI level will be proposed to be used also by the rare diseases community, including open access and data exchange available in EPTRI. Of particular interest will be also the setting up of a EPTRI centralized service dedicated to the identification and further exploitation of project proposal considered relevant to underpin pediatric clinical research. And these activities could be organized in partnership with pediatric initiatives which operate currently in the pediatric clinical trials and studies field. Next slide, please. We have already started contacting and liaising with the biomedical research infrastructure, the ESFRI research infrastructure, and to the same, many activities been already done during the design phase, but our aim is to continue working on this direction and try to really liaise with them, identify activities that we could carry on together with them. And to the same, we would like really to set up a working group with the representatives from ESFRI research infrastructure in order to investigate the opportunity of creating some common services. Our goal would be at the end of the preparatory phase, maybe to prepare a sort of position paper with the aim to set up a framework of collaboration, but also to investigate the respective fields of action and exploit all the available and possible synergies. Next slide, please. Thank you. Some set of activities will also be addressed to the pediatric pharmaceutical and medical device industry and CRO, including SMEs and startup. To the same specific activities will be mainly aimed to tackle actions related to business development in research areas that have a potential to address patients' needs, but also to communicate the project EPTRI to the industrial stakeholders. To the same, we will organize several meetings to identify possible areas of collaboration and business opportunities between EPTRI and all the different industrial stakeholders. And it will also include a strategy on how to best market offers of the transnational services that we will deliver in the preparation phase. And also to attract the interest from industry, several activities will be put in place, like to communicate the portfolio of the transnational access services very regularly, to test the interest of the company in using the services that EPTRI will deliver to the external users through interviews, short questionnaire, or dedicated meeting, but also to present EPTRI in commercial workshops, so in workshops where we can really target the audience we are addressing to. Next slide, please. And very important, so key to the EPTRI development is for sure the engagement with young patients and young people, patient organization, and the public in general, in order to promote the pediatric basic research using a patient-centered approach. This will be very important because it will facilitate a feasible translational research development of the programs of EPTRI, and also ensure that the translational research really focus on community and patient relevant priorities. To the same, the young patients and voice will be instrumental in the research infrastructure development, really from the early start. And in order to ensure that their involvement could be successful and also impactful for the young patients, we would like to set up a dedicated board of the European Patients Forum Young youth group members, which consists of well-trained, highly motivated young patients experts, but also with the young person advisory group, which are groups which are established among young people that are both patient and healthy volunteer, that are linked to academic institutions and children's hospitals. Moreover, we would like also to involve in these activities also the patient representative, families, and patient organization. And our idea is also to work a lot on this direction and to organize and dedicated actions such as organization of roundtables for discussion in order to involve directly patient organization representatives, families in the activities, but also to prepare materials focus on empowerment, education in the field of basic research, and also to involve directly this target in preparing and revising documents in order to be sure that the key messages that we will transfer to them are really understandable for them. So a lot of activities, as you can see from the slide, are have been planned, and we really hope that you will stay on board with us and you can support us in implementing all these communication and outreach strategy in order to let EPTRI grow and become really a fully fledged research infrastructure that can come to all the phases of this research infrastructure life cycle. So, thank you.
Thank you, Mariangela. Wonderful presentation. We can go to the next one with Begonia Nafria from Fondation San Juan de Dio. She will present the EPTRI toolkit for young patients, and this is a very nice one presentation. It will end with a video, so be ready. Pay attention to this very nice presentation from Begonia. Please go. Okay, Begonia, sorry. I'm not sure, maybe I was muted, so I already introduced you. Maybe I was speaking alone. I'm so sorry. So next presentation is from Begonia Nafria, Fondation San Juan de Dio. Wonderful presentation on EPTRI toolkit for young patients. It will end also with a video, so be very attentive. No, we have some issues with Begonia, maybe some technical issues. I'm sorry. So we can go with the next presentation. It is about one of the further areas of interest for EPTRI, where we are working to create another technology research platform. The presentation is about the creation of a medical devices platform. And the speaker is Paul Dimitri from Sheffield Children Foundation Trust. So Paul, the ground is yours.
Thank you very much. I just want to check that you can hear me first. Can you hear me?
Yes. Yes.
Fantastic. Thank you. Okay, so thank you very much for the invitation today to speak about the development of a new technology research platform for on medical devices that has the potential to reach out to multiple centres across Europe. My title slide there says that I'm the director of the National Institute of Health Research, Children and Young People MedTech Cooperative, which I'll speak about in a few minutes. And I'm also the NIHR National Children's Specialty Lead as well. And as has been said, I work at Sheffield Children's Hospital in the United Kingdom. Next slide, please. So our starting point is really to understand the challenges that are facing and have faced the development of child health technology in the last decade. Certainly, one of the major issues has been the lack of targeted health technology for children and young people, and has followed a similar route to in the past to that has happened within the development of drugs for children, which is around about repurposing. So many of the technologies from years ago were repurposed for children from adult technologies, and in a number of cases we've seen suboptimal results, and even worse still, we've seen complications as a result of that repurposing. Another issue is this issue of fragmentation. So what I found, again, around about a decade or so ago, is that many of the technologies that were being developed for children were happening on a localized basis. So there were hospitals or academic units that were developing technologies, but keeping them very much at a local level, and it was difficult to scale up. And that also crossed into industry as well, that our industry partners were developing technologies on a localized level and not really looking at the potential for a greater commercial development of paediatric technologies. And we're in a new landscape now, because children with long-term conditions or rare diseases are surviving for longer. So it's important that we consider the development of technologies not only to improve their healthcare, but to improve their quality of life and their survival longer term. And in that respect, we're going to see improved future opportunities for them. So their opportunity to get greater educational opportunities in their childhood, and also as a consequence of that, greater societal opportunities in the future in terms of working and peer-to-peer relationships. Next slide, please. So we started in 2014, and this, I want to give you a UK perspective. So in 2014, a group of us in Sheffield got together and decided how we would approach the development of a child health technology network on a national basis. And this was founded on the fact that we were doing a lot of development of medical devices and child health technology in Sheffield, but wanted to understand how we could work collaboratively across the paediatric community within the UK to not only to develop technologies, but to ensure that the type of technologies that were developed in different places could be established across the country, as opposed to the previously described localized way. And we received a very small grant of £50,000, and we formed what was called the TITCH network, which still exists today. The TITCH network is Technology Innovation Transforming Child Health. And the TITCH network, established from a number of different NHS hospital partners, together with industry and academic partners, all with the remit of the development and adoption and commercialization of technology designed specifically or developed specifically for pediatrics. How we started was we worked with funders. So we first of all ran some workshops. We started to understand the unmet needs that existed within pediatrics, and then worked with funders to support the commissioning of funding for the focus and development of child health technologies. And if you just press the next button, please. So through that work, we managed to leverage over £6 million of funding for the child health technology development over a 3-year period. What we had also done was manage to establish a national network which had national focus and had become well known not only across the paediatric community, but across the NHS healthcare setting. Next slide, please. So that provided us with an excellent platform to then apply to our National Institute of Health Research to receive funding for a more established paediatric technology network across a number of different specialty areas. So the MedTech and in vitro diagnostic cooperatives within England have been set up to support the development of technology in a number of different specialist areas. There are 11 of these NIHR MedTech cooperatives, and I host the NIHR Children and Young People MedTech Cooperative, which, as you can see from the map there, consists of 7 different theme areas across a number of different specialties, with a cross-cutting theme of transition, which is led by a key opinion leader, an expert in their field, with the focus on developing and supporting the commercialization of technology in those specific theme areas. And that network has been running now since 2018. It's been running for 2.5 years. In combination with the TICH network and NIHR MedTech, we've worked with over 110 SMEs, that we've worked with over 130 clinical experts and over 90 patients and their families in supporting child health technology development. Next slide, please. So from here, the next step is to consider how we develop a dedicated technology research platform on medical devices through the collaboration within EPTRI. So in the green square on the left-hand side, I've already spoken about the specific need to focus on child health technology development in my first slide. What's clear from stakeholder surveys that have taken place through EPTRI in the past is that there is a strong interest in the development of paediatric medical devices. And from that, we also recognize there is a breadth and depth of expertise within that community. And it will be through that combination of experts that will have competencies in the clinical fields of general and specialist pediatrics, pharmaceuticals, different aspects of technology, those that are experts in digital and IT, in artificial intelligence, and other areas in the bioengineering and nanotechnology field that will help support the development of that platform. And it's enormously important that we also work with manufacturers in the commercial sector and regulators to be able to develop a platform to deliver medical devices for pediatrics. So by developing that platform, what we will essentially have is a hub that will link people together and provide the resources required to support the development of pediatric technologies and medical devices, and also that platform to support knowledge transfers with the exchange of new ideas and solutions that will foster research in this field and attract funding focused specifically on medical devices for children. Next slide, please. I've split the needs within this technology platform into 3 areas: development, regulations, and knowledge transfer. So, of course, the need is to develop a platform that will support focus specifically on pediatric medical devices. And actually, what's interesting in this field, having spoken with colleagues that work in adult medicine, is that they see a clear advantage to the development of pediatric medical devices in the versatility that is integrated within that development. And they see that versatility being an advantage in the development of adult medical devices and the repurposing of those devices as well. And it's important that within that development process that we engender a cultural change within institutions and industries such that the development of pediatric medical devices is not seen as either obscure or small, but actually through that networked approach across countries and across Europe provides that level of scale which will allow us to support the commercialization and the commercial opportunity for industry in the field of medical devices for children. Within the needs comes that the focus on understanding the regulations required, and those regulations will vary across multiple different countries, and they will also vary between the development of adult medical devices and pediatric medical devices. So it's important that we gain an understanding across Europe as to the regulations which exist, and that the technology platform becomes a reference point for those academics and industry partners who need access to those regulations and expertise to support the development of their medical devices. And fundamental to that platform is knowledge transfer, which I mentioned in my previous slide. It's important that we facilitate medical device research collaboration, support the development of medical devices and other technologies, and that we provide dedicated training and education in this field of pediatric medical devices through training programs developed through EPTRI. Next slide, please. So in terms of the steps for developing the technology research platform, I've divided this into the who, what, and how. And this is defined by structural capability, process capability, and relational capability. So it's within the structural capability, the who, it's important that we identify key experts in the paediatric medical device field who can work collaboratively through research projects and through commercialization opportunities to support the development of paediatric medical devices. And how we can do that is establish a network of partners from the clinical, industry and academic fields, but importantly as well to incorporate representatives from the patient and family groups to ensure that we develop an established network to support paediatric medical device development. And the question I've asked there is, could this potentially be a network of networks? So one of the areas of scoping that we need to understand is whether we are talking about institutional collaboration or whether, as we have done within the UK, is establish a network, and we are linking multiple networks across Europe to create a network of networks that can support the development of medical devices across Europe. Our surveys will help to establish unmet needs, and it's important that we find commonality across those unmet needs to support research in this particular field. And those unmet needs will come from researchers, and they will come from patients and other collaborators within this platform. Once we've established an EPTRI platform for medical devices, what this will then do is foster medical device research in this field. And it's important to do that we collaborate with funders to establish funding streams and also to create a funding roadmap that allows us to identify areas of opportunity to fund paediatric device research. Next slide, please. So the platform actions are split into three areas. Clearly, we need to establish short, medium and long-term strategic plans that are driven by aims and objectives, and that will be developed through the platform activities. We'll need an established research strategy based upon the unmet needs that have been identified through collaborative workshops and surveys supported through academia, clinicians, industry and patients and their families. And it's important as well that we have a regulatory framework strategy, as mentioned previously, such that we understand the potential barriers and requirements in different countries to develop paediatric medical devices and to support commercialization within and across Europe. Next slide, please. The objectives of the platform development are to identify centres with expertise for development of child health technology, and that must incorporate design as well as those with specific expertise across the technology areas. And what I suggest we do is that we take an innovation pathway approach, such that we look at expertise that support development of technology, prototype analysis, validation, and then endpoint user and usability assessment to ensure that we have not only accessibility to the expertise, but acceptability to the technologies that are developed. And I propose that we develop a three-dimensional institutional CV, which I'll talk about in the next two slides. I've mentioned this earlier, but we must foster, sorry, previous slide again, please. Thank you. We must ensure that we foster paediatric research in this particular field, and that we engender knowledge transfer through collaboration between medical device experts across different parts of the landscape and field, and that we support authorization and regulatory processes that should include CE marking. We need to develop a funding roadmap for research, and we also need to develop an education program for child health technology and medical devices. Next slide, please. So in terms of developing this three-dimensional platform CV, is that we gain an understanding of the range of expertise, which will include multiple technologies, as I've demonstrated in that diagram below, and that we do this across the innovation pathway. So through surveys, we'll understand expertise in technology development, and that what that will allow us to do is to develop a pan-European profile in medical devices for children, and this three-dimensional platform CV. And this CV has to be both patient and industry facing, as well as an internal CV for us to understand where the expertise are. It will also create an open space for collaboration across complementary specialist areas. Next slide, please. And through that scoping exercise to support the development of a three-dimensional CV, we need to look across the various parts of the innovation pipeline, right the way from ideas to proof of concept towards the end stage of commercialization, diffusion and adoption, not only to understand the scope of institutional expertise in the fields of technology, regulation and design, but also to understand the scope of that institutional expertise across that innovation pipeline. And this is what I mean by three-dimensional, that it's across the innovation pipeline. So we have a breadth of expertise across digital, through the technology areas, but also a depth of expertise across these different areas within the innovation pipeline. Next slide, please. How we define these unmet needs is that we will support workshops and surveys that will define future work streams, and that we'll be able to collate known areas of unmet need across European centres to be able to define these common themes. What we then need to do as the next step of this process is to align those unmet needs with the funding opportunities that we've identified with that funding roadmap, and then support the development of an educational framework that will help us to develop areas of expertise within the specific work streams to support future research opportunities. And fundamental to this is the centralized coordination of each of these work packages and work streams. Next slide, please. My final slide is really a message about how we think about medical device development for children and young people. There is clearly a great opportunity here across Europe to support collaboration between those working in academic institutions and networks, those working with industries that will include SMEs, and those working in their respective clinical fields. But fundamental to technology development across all parts of the innovation pipeline is to make sure that we involve patients and their families to ensure the wholesale development of paediatric medical devices from proof of concept right the way through to commercialization and adoption. So it's important with this platform that we make sure that we have the appropriate PPI input through patients and families to support medical device development. Next slide, please. So summing up, our aim in this relatively early stage is to develop a pan-European technology research platform for medical devices that will cut across clinical areas, academia, and industry to support child health technology development, that it will be underpinned by a strategic framework fundamental to defining and developing this platform, and that we will do this by aligning expertise with unmet needs, and then aligning that with a funding roadmap across the innovation pathway to support research in this area. That the ethos of this medical device platform is to support user-centered innovation through true participation with patients and their families, and that this platform becomes an open space for the cooperation and exchange of knowledge for medical device experts through the EPTRI initiative. Thank you very much.
Thank you, Paul. Very, very interesting presentation. So you have detailed plan for establishment of a new technology research platform. It will be composed of several parts, and we have a second presentation from Maria Henke from University of Lübeck describing their open lab for robotics and imaging in industry medicine that will be also tailored to pediatric applications. So please, Maria, the ground is yours.
Thank you very much for the introduction. Can you hear me?
Yes, very well.
Thank you. Yes, so now it will be a little bit more technical. I will give you an introduction into the possibilities we have in our open lab, and show you some projects, and also show you some projects which are related to pediatric medical devices. Next slide, please. So the Open Lab is a research platform and is a joint institution of two institutes of the University of Lübeck, the Institute for Robotics and Cognitive Systems and the Institute of Medical Devices. And our mission is to offer access to our expertise and equipment to tackle technical challenges. And we aim to develop individual solutions which center on the patient. And we have several possibilities in terms of our equipment, which you see on the next slide. So we have nearly 10, no, more than 10 different robots available. We have different tracking systems in our labs, very specialized measurement devices, different kinds of 3D printers, and different kinds of imaging systems, of which the MRI and the CT are small volume CTs. And we also have the possibility to use an X-ray C-arm, and we have different ultrasound machines available, and different high performance cameras. And on the next slide, I introduce our services to you. So the OLRIM is the short name of this very long open lab for robotics in industry and imaging in industry and medicine. You see how long this is. So we offer the service that we develop software, that we do prototyping, or that we use within collaborations with the local, at the moment only local industry. They use our robotics hardware, our imaging hardware, and also our research facilities. And for sure, we offer scientific services, and the main part is in the combination of imaging and robotics and artificial intelligence applications. And on the next slide, you see a short illustration from our Mirana project. You can see this little robot, and which is running into the hospital. So he needs an intelligent path planning, and he navigates. So it navigates. So you see, I call him he, although it's a thing. He has got a chatbot available, and he can detect persons, and he is able to guide persons through the rooms without losing this person. So there's the detection of persons cares that the person doesn't get lost, and there's a global navigation implemented. So this mobile intelligent robotic agent can serve as a personal guide, as you have seen in this video here, and can also be used as an info point. And in the other in the talk we've seen before, there was an image of the Pepper robot, which looks a little bit like a human, and we hope that this small robot, which is small and looks like a robot, but it's also small and cute, is also something which is accepted by children, because this other thing which looks a little bit like a human but is not a human and doesn't act human, may also cause fear in the little patients. So this is something we are looking forward in the based on the results of the Mirana project. And in this slide, you can see the two examples of our projects. On the upper part of the slide, you see a robot holding the ultrasound probe of the ultrasound machine, which is on the right-hand side. And in the middle, there's the tracking camera, which gives us the location of the ultrasound probe in the room and in relation to the patient. So this is one example, and on the bottom you can see the production or the result of the 3D printing of a model based on CT images. And on the next slide, I've brought you two examples. On the left, you see our own work. This is the first test of the idea to use ultrasound in water to examine the forearm. And this is especially important. So in this case, you see that this first experiments were made with adults, but the application is especially relevant for children because the fractures in the forearm are not so seldom, and the radiologists try to avoid CT imaging and they try to avoid X-ray imaging, and thus children with a suspected forearm fraction have often to go into the magnetic resonance imaging, and this means that they also this requires often sedating the patient, which is another burden for this small body. And so this work is the start of the possibility to diagnose forearm fractions with ultrasound, and so own work on the left-hand side, and on the right-hand side you can see another publication in which the idea was tested to have this with a not with handheld by a person, but with an automatic ultrasound image, which is taken here. So there are some groups working on this, and we are also talking to the hospital, which is located on the same ground as the university is, to see how we can drive this project further. So this was a little more technical view into the pediatric device, medical device research, and I thank you for your attention.
Thanks a lot, Maria. Very interesting field to be developed for pediatric research purposes. Now, Begonia is ready to start with her presentation on EPTRI Toolkit for young patients. Please, Begonia.
Hello, good afternoon to everybody.
Yes, very well, thank you.
Hello, it's my pleasure to introduce this deliverable of the project. We have been talking this afternoon about medical devices, robotics, and also in these previous presentations you have mentioned which the role of the patients, that it's important and essential in our work. But obviously, patients, and this is the approach of the EPTRI initiative, patients need to be around and obviously in the basic research. This is the main idea of this toolkit, to educate the patients about which are the roles and the activities in what they can be involved when we are talking about basic research. With this idea in mind, we thought that the best format and the best way to educate and to inform the patients will be in a multimedia format, explain the different dimensions in which they can contribute, and also show them an example about a potential real case to show them which is the role that they can have and how important is for the activity that they can be involved. Say this, if we move to the next slide, you will see the objectives that we envision at the very beginning of the design of this toolkit. The first one, as I said, it's to educate the patients and the families. We think that it's a social commitment to educate them, because this is not the best way to ensure that they have the knowledge and the capabilities to be engaged in our activities. The second one is to inform the general population about the basic research and obviously in the scope of pediatric medicine's development. The toolkit, it's addressed to patients and families, but the same information is accessible for the general population, and still there is a big gap there to know what is the work that we do in basic research before we move to the clinical research. The third objective is to create a collection of different resources that we think that are essential to educate and inform in the different dimensions that afterwards I will explain. It's not only to provide a multimedia resource with the general information about what is the role of the patients in our field. For us, it's very important to curate and make a great selection of resources for the people that it's interested to know more in depth about these different dimensions. The toolkit has a general video with this example that I mentioned to you. And obviously, the idea is that this toolkit will be a guidance and a resource that the people working in basic research can use to educate the patients and the families connected with the area in which they are working, to educate them and facilitate their active role in the different projects in what they are involved. This is the idea, and we think that this tool needs to be disseminated as much we can. And obviously, it's accessible in the website of the EPTRI project, and you are free to disseminate and share this information with all the people that it's connected with your projects and your social media, website, etc. If we move forward to the next slide, you will see which is the look and feel. It's a look and feel very modern. A look and feel basically focus also in the target of this toolkit, that beyond not to be addressed, as I said, to the general population, also it's addressed to the pediatric population. At this, and in this target, you need to work with a very fresh look and feel, innovative, and something that can attract their attention to know more about this field. If we move to the next slide, another more. The idea of the toolkit, as I said, it's to bring a general video. It's like a story with an example about an oncology disease. You will see which is the journey from the discovery of a potential treatment in the lab, and also at the end of this journey when this treatment can arrive as a clinical trial to the patients. In the bottom of the toolkit that it's accessible in the section about resources and media in the EPTRI website, do you have 7 areas of knowledge with these resources that I mentioned that we curated. If we move to the next slide, you will see this list of the different areas. We selected and curated resources to explain what is basic research, also to gather information about groups and lines of research, introduce the concept of translational research, the donation of patient samples, because in some cases here is the moment and the point where when the patients start to be involved, and when they start to know that this is a very meaningful and essential contribution that they can have. introduce them in the field of animal testing and all the topics that are around about this experimental area. Obviously explain which is the concept about patient and public involvement, because this is a very technical wording, but sometimes even expert patients, they don't know which is behind of this concept, in which activities they can contribute, because for us, no, the role of the patient is a cross-cutting approach. They can be and they need to be from the very beginning of the journey. It means that in some cases also they can help to prioritize needs in the discussions with the research teams. But obviously the journey, it's long. We know that there are many years, no, after we can have a treatment approved in the market. And here in this section, we create resources to explain the different activities in which they can be involved. We have removed from this topic the concept of fundraising, because you know that in the literature you can read sometimes that when we talk about basic research, basically PPI is understood as a donation of patient samples and a donation of resources for research. And for us means something beyond to provide resources, even if are biological or not. For us, PPI is the real voice that needs to be heard and can shape the basic research. For this idea, we split these topics in three. And the last one is about ethics. The patients and in general the society, they need to know that ethics is behind of every research activities, and they need to know which are the principles that we have in our initiatives and how they can use and exercise their rights. We have here resources in different languages. The toolkit, the general video that we will see after this presentation, it's in English, and we are at this moment finalizing the process to include other European languages. If anyone in the call is interested to support us in the process to add a new language to the toolkit beyond the English, French, Italian, Spanish, etc., we are going to be very happy to add it, because as much languages and more accessible for everybody this toolkit, the better to educate and inform the patients about which is their active role in basic research. Next slide. This is the general look and feel of the toolkit. As I said, there is this general video that once you access to one of these 7 areas of topics that I have mentioned previously, you have also different subcategories about resources. Here you can find from other videos, guidances, or even links to websites that we consider that are interesting or are referenced at the European level. Next slide. Okay, the next one is the video. Let's see if we can hear the audio. And this is the premiere for all the people that it's in this conference of the video. Thank you.
Like in every other scientific area, researchers and healthcare professionals work based on scientific evidence, which is information or data obtained from scientific experiments and verified by the scientific community. Doing research in this field must take into consideration children's peculiarities. Children are very different from adults, mainly because their bodies are developing and constantly changing. Basic research sows the seeds to develop new solutions for future medical use. But how does basic research build the foundations for these discoveries? Let's follow a case together. A treatment for the most common eye cancer in children, retinoblastoma, which involves the use of a genetically modified virus. First, different research groups from several countries join together to communicate other researchers' a promising finding: the use of certain viruses as a therapy against cancer. Several studies have been showing in a conclusive way the possibility of modifying some types of virus to get rid of carcinogenic cells. A translational research is this option, and to do that, who has the expertise to modify the virus. A a biotechnology group takes the first step to carry out studies based on the knowledge of basic research groups. In this case, they have modified a virus which might now only attack cancer cells. The next step is to test it on biological models. With the help of the patients and their families, who donate samples of body tissues, researchers reproduce the encounter of the modified virus with the tumor at a microscopic level. It lays the foundations for the next step, testing in small animals. The accomplishments of the different research groups and the publication of their results make it possible to successfully try the potential therapy on children suffering from this type of eye cancer. A clinical trial can now be conducted. If the outcomes of the clinical trial are positive, patients gain access to a less invasive potential cure and with fewer side effects than previously available options. Nowadays, and thanks to the collaboration of researchers, clinicians, patients, and families, basic research in retinoblastoma has allowed the design of the first clinical trial to potentially treat this disease. To continue having a positive impact on children's lives, we need to involve them and their families in the whole research process. Together, we can encourage patient-centric research in pediatrics. We must help others.
Thank you, Begonia, for this delicious video. I don't know if you want to add anything else.
No,
Anyway,
it's all from my part. Yeah.
Thank you.
All
Thank
you
you,
mentioned
Begonia.
Okay, thank you.
Thanks a lot. Now I'm going to introduce another new and innovative field of research that will lead to the development of a new platform dedicated to placental research services. So I'm very happy. We have two presentations on this. I'm very happy to introduce František Stod from Charles University. Please, František, the ground is yours. Yes, we are ready with your presentation. Okay.
Thank you very much, Donato. Just to confirm, can you hear me? Can you see the presentation?
Yes, very well. You can go.
Perfect. So once again, thank you very much for inviting me. And as you said, the topic of my talk is introducing this new placenta platform, which is actually a very new addition to this EPTRI initiative. We started talking about this earlier this year. And it's a thematic research platform dedicated to placenta studies under EPTRI, with the aim being to fill gaps in developing and providing know-how, technologies, and access to many in vitro, in situ, in vivo models of research on placenta physiology, pathology, and everything. So my job today is just to introduce who we are, what we do, and why we do it. So this is the structure of the presentation. I will just introduce quickly current team. I will try to stress out the importance of studying placenta, and then I will introduce and guide you through the method that we currently have available. I'll start off with a team that consists of four teams, actually European and Canadian teams. Professor Christian Albrecht from Switzerland, Kathy Wallencourt from Canada, Udo Market, who is now here with us and will continue the talk after me, from Jena University in Germany, and myself, Charles University in Czech Republic. All the team members are people with decades-long experience in placenta research, and we are actually at the very beginning. So I hope this platform will grow and will include more European or transatlantic groups as well. Now, why to study placental physiology, pharmacology, toxicology? Because actually EPTRI is focusing on pediatric research, which is kind of postnatal period when the child is born, and including placenta research into this EPTRI activity actually in a way stretches the research activities beyond this horizon to even the prenatal period before delivery. And as you can read here, prenatal period is the foundation for later life outcomes. This has been published several times and confirmed. And the thing is that actually during pregnancy, the in utero conditions have to be maintained in optimal state to make sure that the fetus is properly developed and programmed for future life. And placenta is the unique organ that takes care of it, that maintains optimal in utero conditions. I believe we all know that if something goes wrong, if there is some placenta malfunction, we can see some acute effects. For example, here you can see in the picture this infamous thalidomide disaster, or bottom spina bifida teratogenic effect of valproic acid used in pregnancy, or very small babies due to fetal growth restriction. And these are, in a way, acute effects that we can see straight in the delivery room. When the baby is born, we see something is not correct. What maybe population and people are not aware of is that even when the baby is born happy and smiling and healthy, it may develop some diseases later in life due to placenta misfunction. And these diseases later in life are typically metabolic diseases such as diabetes mellitus, cardiovascular diseases, or psychological diseases such as autism, depression, schizophrenia. And this is something we call fetal programming, or developmental origins of health and disease. I will devote one more slide to this DOHAT because it's really actually a new field of research, and it really includes placenta in the middle. And the story goes very quickly in this line that if there is a woman, pregnant woman, that suffers some pregnancy complications such as malnutrition, some inflammation, is treated with some drugs, or suffers other diseases, then the baby, despite being born healthy and fine, can grow, and at the age of 30 or even later, 40 years, the offsprings have a high tendency to develop diseases such as CNS disorders, pulmonary hypertension, as you can see in the picture. And this connection actually between the pregnancy complications and diseases later in life, they have been clearly confirmed by epidemiological studies, large-scale epidemiological studies make these links very straightforward. But we don't know what's going on in the in between. Definitely, we know there is placenta for this 9-month pregnancy that is supposed to take care of the baby development programming, and there are many possible targets that can be affected by pregnancy complications and actually misprogram the baby. So although we do not know the exact mechanistic or molecular explanations, we can definitely blame placenta for creating not optimal conditions in utero, leading to improper fetal programming and development of diseases later in life. So that's actually why we want to, or why we have to study and understand placenta as much as possible. Because placenta is actually a very complex organ with many functions, including endocrine functions, just for this, for example, fetal programming. And I need to stress out that it's really difficult to investigate placenta due to many reasons, ethical reasons, technical reasons. And today we have no single method that will answer all our questions. So nowadays we have to really rely and perform on several methods, experimental methods, and treat the results in a complex manner. So these methods, we can briefly split them into in vitro cell-based methods, organ perfusions, ex vivo human placenta-based methods, or in vivo imaging techniques. And this is actually why we are putting this placenta platform together, because the unique feature of this platform and the strength of this forming platform is its capacity to offer integrated services by these partners in a hierarchical cascade, starting with low-cost, high-throughput analysis, followed by some more complicated ex vivo tissue explants, animal experiments, or up to ex vivo placenta perfusions. And so this strategy is in a way to avoid cost-intensive analysis. So I'll briefly guide you through the methods that we have available within our platform at this moment. And I'll be really quick because we don't have much time to explain all this method, but feel free to ask or email me with questions. Of course, we can start with in a way, inexpensive in vitro screening methods based on cells, transduced cells, or placenta-derived cells. And this gives us a very quick and inexpensive picture of drug transport, nutrient transport across the placenta, because we can grow these cells in a polarized manner on a membrane and treat them with substrate and see the accumulation of efflux. We can test substrates and inhibitors. For example, we have used this method as a screening method for antiretroviral therapy of pregnant women, HIV-positive pregnant women, and we need to treat them with more antiretroviral compounds to see their own interactions in the placenta interface. From this, we can move to more complicated models of animal placenta perfusions. Nowadays, we have in the world, there are two teams working with perfused animal placentas. One is in California, Oregonian. Second one is here in my faculty in Czech Republic. And although this is just an animal model, it correlates very nicely with human placenta in many aspects. We have proved this and described this on several occasions. So again, this is actually another model in our portfolio. We, of course, need to work with human placenta as well to get closer to in vivo human conditions. And for this, we have several methods. For example, these relatively easy and straightforward methods, such as fresh villus fragments, placenta explants, placenta homogenates, to study again placenta physiology, toxicity. And Udo Market will continue, and he will talk about placenta explants and placenta toxicology. So I'll move to more complicated methods again to study placental physiology and toxicology. And this is very sophisticated method of membrane vesicles isolated from human term placenta. And actually we can isolate these membranes and vesicles from both maternal side and the fetal side of the placenta. And then we can, in the lab, we can really investigate what's going on the maternal side and the fetal side of placenta and get very clear picture of, for example, homeostasis of hormones or nutrients in placenta. Probably the most complicated or demanding, laborious, expensive method is this dually perfused human term placental local system that is also available in our platform in Jena and in Bern. And again, this is these all methods are very, in a way, nice to study drug or nutrient transport across the placenta, to study placental homeostasis of endogenous hormones, to study placental endocrine function, glucocorticoids, monoamines, and which is something we are now actually using in the placenta platform. Then we also have to be available to provide in vivo models for pregnancy or placenta research. Of course, we at this moment we can use only animals for example, chronic exposure of animals to pharmacotherapy or to some pathological conditions. We can subject them to stress, inflammation. We can treat the animals with drugs that are commonly used in pregnancy, such as antiepileptics, antiretrovirals, antidepressants, and see the effect on placenta physiology and placenta functions. We can also offer and perform basic pharmacokinetic, pharmacodynamic studies in pregnant animal models. And we are also working right now on establishing a beautiful kind of innovative imaging system to study fetal placenta development. I'm not sure you will see the videos here, but we can actually using this ultrasound Doppler machine, we can investigate perfusion of placenta, blood perfusion of placenta from both maternal and fetal side, and again subject the animals to some pathological conditions that are resembling human conditions, or treat them with some drugs, pharmaceuticals during pregnancy, and see again the effect on placental vasculature, placental development, fetal development. So these are actually, this is the portfolio of methods that we managed to put together at this moment, but we are still not giving up. We are actually continuing working together. And this is, I'm really happy to announce, this is result of last week, where we applied for a grant with Christian Albrecht from Switzerland. We applied for a grant to be able to engineer a novel cell-based model for assessing maternal-fetal drug transfer during pregnancy. The grant was approved last week. So this is just to say that although we do have a huge battery of several models to study placenta, we are continuing and trying to expand to be able to provide as much as we can. And I believe this is all from my side. So I hope this placenta platform will prove viable and important for EPTRI initiative. I hope it will grow. And at this moment, I thank you for your attention on behalf of all the members of placenta platform. Thank you.
Thank you, František. Thanks a lot. Now I'm giving the presenter to Udo Market, University of Jena, and it's a practical example on a project and on the possible interaction with the EPTRI. The project is PLATOX, Placenta Toxicology, Human Placental Explants for Drug Testing. Please, Udo, the ground is yours. We cannot hear you. Udo?
Yes, now you can hear me?
Okay, yes.
Okay, I hear my echo. Now it's better, yeah. Okay, so sorry for this problem. Thank you for the nice introduction. Good afternoon to everybody. I'm going to present you part of PLATOX. PLATOX is a project which we have submitted to the German Ministry of Research and Science a couple of months ago, and we hope very soon for a result, for a positive answer, and hope that this will be sponsored. Oops. I am originally located here at the University Hospital Jena, and this project is in cooperation with Professor Andreas Schober from the Technical University Ilmenau in our region, and Gregor Schlinghoff and André Schmidt. So human placenta, we have heard about it from František Stout, and I have some short introductive parameters here. So the human placenta is the barrier between mother and embryo or fetus. It has a bidirectional selective transport. So that means substances can pass from the mother to the fetus or from the fetus to the mother. It is a very important hormone producer during pregnancy. It is very important for immunoregulation because the maternal immune system has to adapt to the presence of the allogenic or semi-allogenic fetus, which has genes also from the father. So the placenta needs to prepare the maternal organism for this object, for the fetus, which is not known to the mother, and usually an immune reaction should be expected. So the placenta is a very natural organ. It's unique in humans. You can receive it freshly every day in many places, and it's complete, it's vital. Compared to other organs, it offers enormous advantages and can be used for toxic analysis, not only in regard to the placenta itself. It's simply human tissue, which is vital and fresh. So if you compare other organs derived from pathology or from surgery, because either the patient has died or because the organ is sick, but usually a healthy organ is very rare to receive from a human. Yeah, we have an enormous species difference. So in animals, you can do very important, very nice long-term exposures. In the human placenta, it's usually a short-term exposure, but it's human. And the placenta is an organ which differs strongly between the different species. So the models, we have heard about them. There are different models, cells. You can isolate them from a placenta, or you can use cell lines. You can culture them in 2D or 3D models. You can use whole placenta cotyledons, for example, for placenta perfusion. The cotyledon is a kind of a lobe. It's a lobe organ like the lung placenta. You can use placenta explants. We have also heard about it in the previous talk. These are fragments of the placenta. Yeah, and to use these fragments, these explants, is the aim of the project PLATOX. So I show you here, this is a very old image, but it's still valid from the placenta. This is a cotyledon, which has, this is the fetal part here. This is the maternal part. And here you see an artery and a vein, which serve here to one villus structure. And this structure is called a cotyledon. So here we have another cotyledon. And this can be perfused. This is also what František has shown you. We can insert needles into the artery and the vein and perfuse these vessels here. And we can insert needles here into the intervillus space and perfuse from the maternal side also. So alternatively, we can cut here little pieces, explants, and culture them. So this is how it looks like. You see here the placenta. You see here, this is for placenta perfusion. You see here needles installed into the artery and vein on the fetal side. This is how the machine looks like, the system for ex vivo placenta perfusion. Lots of little tubes and chambers and pumps. And we can test transfer of substances from both sides, from the fetus to the mother or from the mother to the fetus. We can test accumulation in the tissue, and we can test any effects on the tissue with this method. This is the other side. This is the maternal side of the placenta. There it's impossible to find or to define blood vessels. It's just a bloody surface, and there we have to install the needles carefully through the tissue into the intervillus space. And this is the whole system. You see water baths to warm up. There are oxygenators, pumps. It's a quite complicated system. So the different placenta models, we have heard about them. We have, for example, cell lines. Exposure time is unlimited. Availability is unlimited. Reproducibility is rather high. Workload low. Then we have cells, isolated cells, for example. They survive usually only a few days after isolation from placenta. Availability limited. One-side perfusion or dual-side placenta perfusion, what I've shown you, maximum of 8 hours. Availability more or less maximum of 3 per week if you work quickly, so 3 placentas per week. So you see here is quite a big difference. In cell lines, of course, you can do hundreds of experiments, and here you are very limited. So the aim now of Platox is to use the placenta explants and to improve this system. And you see here the aim is to culture them at least for 2 weeks and to have at least approximately 100 samples per placenta taken simultaneously from one placenta. This is where we culture them in. So you see here, first here on the right side, a so-called MatriGrid, which is a culture structure for 3-dimensional cell cultures. This is patented by our partners of the University of Ilmenau. But we saw this was not good for this villus, very sensitive villus structure of the placenta, and they have continued developing a system which is now called the TISGrid, also patented by the same group. And we have cultured villus tissue here for 4 weeks, and it looks vital, or mostly vital after 2 weeks, as shown by the green staining here. And this can be inserted, for example, in these disposable bioreactors for perfusion. And in this system, there's a superperfusion. That means the medium does not flow through the tissue, it flows over the tissue. Here we have tested, for example, estradiol, so one of the major hormones produced by placenta tissue. In our cultures, you see here, this is 14 days. And when we use this superfusion system with the bioreactors, we had, after 2 weeks, we had the highest estradiol production, which means the most vital and most relevant tissue. So this encouraged us to work further on these systems, on the superfusion bioreactors. So this is how we do it currently. It's handwork. So we have the placenta, we cut pieces out of the placenta, and always smaller and smaller and smaller. So we put them in medium, and then we are able to weight them. And usually we use very small pieces, like 20 milligrams of the placenta, and culture them in plates. But you can imagine, if you have all this handwork, it's very difficult to do it quickly. So it's must be done carefully. And when you want to prepare 100 samples, for example, it takes time. And the first sample and the sample number 100 is different just because of the handling and the loss of time. So therefore, we have designed this project, and this includes especially an automatized punch. That means it's important to take 100 samples or 96 samples simultaneously from the placenta and transfer them simultaneously in a plate. So currently we are working with these 24 well plates, but the idea of this project is to expand that. And here in these plates we have these tissue grids, and we can superfuse them in these plates. So our partner in Ilmenau, they produce plates where every single well can be superfused and controlled individually. And yeah, this should help to standardize and to speed up this method. Yeah, so all common after explant culture, after 2 weeks, we should be able to do all common laboratory methods to assess toxicity of test substances. So this is the question. We put the substance on these cultures in different concentrations or different times, and then after this incubation, you still have tissue, and you can do a set of different analysis, whatever you want to do in the laboratory, immunohistochemistry, PCR, any omics, sequencing, and so on. You can do immunodetection, like Western blots, and so on. Yeah, and you can test cytotoxicity of these test substances, hormonal disruption, inflammation reactions, metabolic changes. These test substances can be drugs, can be environmental toxicants, additives to nutrients, pharmaceuticals, cosmetics, and so on. So this is the aim of the Platox project to standardize this sampling from the placenta for this toxicity assessment. And this is also the end of my presentation. Thank you for your attention. And I'm not sure if we have time for questions or afterwards. Okay, thank you very much.
Thanks a lot. We have no time now for questions, but we will try to have all the questions at the end if there will remain enough time. So now I'm glad to introduce Daniela Nica. Her presentation is an example of the EPTRI presence in countries that are not member of the European Union. She's representing the University Hospital Mother Teresa, which have been delegated by the Ministry of Health of Albania to represent the country in the current project. And so, Daniela, the ground is yours.
Good evening. Do you hear me, Donato?
Yes, very well, Daniela. Please
yes,
go
on. Now, okay. Good evening, everybody. Dear Donato, I want first to thank you for giving me the opportunity or the possibility to be part of this very interesting webinar, when I can learn about many researches and surveys from other colleagues, part of EPTRI, and also represent the situation in Albania. As you said previously, Albania is not a European, let's say, part of European Union country, but it's an associated one. And we are trying to support the participation of Albanian institutions in the research community and in the enhancement of the pediatric research. In Albania, there are three main institutions part of EPTRI. The first one is the Ministry of Health and Social Protection. And then it's the University Hospital Center Mother Teresa, which I do represent here. And also a new institution, part of this is the National Agency of Scientific Research and Innovation. All these three institutions have expressed a high interest and support for this new European research infrastructure, as it is of a high relevance for research community. Next slide, please. Yes, okay. The Albanian Ministry of Health and Social Protection has supports EPTRI with the political letter of support in July 25th, 2019. And in this letter, it has recognized the need of the transnational research for pediatric medicines research and the relevance of EPTRI for the development strategies of Albania in the field of health research. Next, please. Meanwhile, the other institution involved is the Tirana University Hospital Center Mother Teresa, which I represent, and where is concentrated the biggest, let's say, team supporting EPTRI in Albania. Our hospital center, it's a university one, and is the biggest health institution in Albania that offers tertiary care, health service, let's say, teaching and academic research. And we are involved in different activities. We are a partner project in EPTRI, mainly responsible for the identification of the research units and their aggregations in Albania, including the mapping of therapeutic areas and the reference groups in the EPTRI fields of interest. Another activity is that our hospital center contributed in the feasibility study to assess the validity of concepts developed in terms of support from the member states government. And the third, let's say, activity is that our hospital center has been included in the recently presented project proposal EPTRI IA. So many of you knows what project is this. As I told you, the third institution part of EPTRI is the National Agency of Scientific Research and Innovation. You can see there the let's say the old name, because till 2016 it is changed. But anyway, it is a legal institution that facilitates the exchanging of knowledge, mutual activities, and partnership with and outside, within and outside the country. We call it briefly NASRI, so the National Agency of Scientific Research and Innovation, is a coordinating and guiding structure which cooperates with institutions in the field of science and technology for sustainable development of the country in line with national priorities, development of scientific and technological policies, and management of research and development institutions. The agency has been included in the EPTRI memorandum, in the EPTRI memorandum of understanding, and it is currently working on the provision of to EPTRI with a letter of financial commitments. Next, please. One of the surveys that our hospital center has participated is the survey regarding the oral formulations. This survey aimed to capture the perceived preference of oral dosage forms in children, depending if they have taken or not one, and what type of oral formulations. And as you can see in the blue color, in Albania, let's say that solid formulations were very preferred by the oldest children, let's say. And meanwhile, the liquids were preferred in the small ones. This was based, the most, let's say, important factor that influenced the preference is taste and swallowability of liquid formulations. It was very nice because this survey was available in local language, and it was filled by many Albanian children. Next, please. Another survey was where our hospital center participated, was medical device survey. This was also, let's say, part of EPTRI, the European Paediatric Formulation Initiative. And when Albania participated in this short questionnaire to understand what devices the young patients and their families use more, and help finding out the ways of making the devices more user-friendly. In this survey was addressed, and is still open, let's say, is addressed to boys and girls between 10 and 18 years old, and it is also in Albanian language. It is important that the children express their knowledge and their feelings in these questionnaires, and show us that how these devices are used, if they are used properly, to make sure the correct dose of medicines are taken by them. The survey will help to find what devices the children use and how easy they are to handle, to help find ways of making the devices more user-friendly. Next, please. And at the end, let's say that the expected impacts are that we would like to be part of a distributed European research infrastructure able to better coordinate and foster cooperative efforts regarding existing expertise in the pediatric research. And also to reduce fragmentation of the pediatric research landscape through harmonization of procedures, implementation of common standards, and promotion of scientific excellence. Another expected impact is to enhance the pediatric research and to implement the key enabling technologies for drug development. And at the end, to improve, of course, the main impact is to improve the health conditions of children. That is the presentation by my side. Of course, Albania, as I told, is a small country, and for now, these were the possibilities, our possibilities to participate and to develop this kind of activities. Thank you.
Thanks a lot, Daniela. Your great effort is really appreciated.